So, coming back to the properties of the bone, in the last module I have discussed that how to define the fracture related properties, particularly the fracture toughness, that is that critical stress intensity factor under mode 1 loading. So, this module will discuss some of the properties and will try to understand that how the bone properties depend on the mineral content or bone properties can vary from patient to patient. So, this particular slide essentially plots the fracture toughness values of the human cortical bone measured using single edge notch beam technique. And what you notice here is that this along x axis the age of different patients from which these cadaver bones were taken, and then they measured the fracture toughness are plotted. So, there has been a general trend that higher the age of a patient, there is a general decrease. So, there is a decrease in the fracture toughness with increase in age. That means older patients, the bones in older patients will have more proneness towards the fracture, or the older patients will be more prone to bone related fracture than the younger patients. The other thing that what you notice, that window over which this fracture toughness of that human cortical bone, that they vary, and this window is somewhere 4 to 7 MPa square root meter. So, this is the window across which this cortical bone fracture toughness is. So, if any material which you need to use that as a bone replacement materials, if their fracture toughness somehow can be within this particular window 4 to 7 MPa square root meter, although larger is the fracture toughness, better it is. However, 4 to 7 MPa square root meter, that is the typical cortical bone fracture toughness, and that is what one has to match with that kind of natural bone. Now, in terms of bone density, as I mentioned earlier, that calcium phosphate content in natural bone decides the bone density. So, larger the bone density means that this bone is much more healthy and adult bone. Lower the bone density means the bone has much lower calcium phosphate content or hydroxyapatite content, and typically this lower bone density is observed with older patients. Again, there is a very linear trend with increase in the relative density of the bone, that maximum fracture toughness also increases. It is a log scale 0.01, 0.1, 110. So, therefore, it must be somewhere between 4 or 5 to more than so this is the typically that cancellous bone structure. So, cancellous bone is much weaker. So, cortical bone has a fracture toughness between 4 and 7, here cancellous bone has a fracture toughness between 0.1 to 1 MPa square root meter. So, essentially the cortical bone cancellous bone has extremely weak in as far as the mechanical property is concerned, and therefore, it has a extremely low fracture toughness. So, it is extremely fragile as far as the mechanical loading is concerned. So, these two bone, cortical bone and cancellous bone, they have a widely different fracture resistance properties in terms of the fracture toughness, and also they show bone mineral density dependent fracture resistance properties. And in both the cases you can see in the cortical bone also that Kc increases as the bone mineral density increases, bone mineral density increases. So, because if you look at the age in the going to the right hand side, if you plot the bone mineral density, that should go on the left hand side. So, larger is the bone mineral density, more is the fracture toughness, and here again you can see that more is the bone mineral density, more is the fracture toughness. This is also true for cancellous bone. However, the magnitude of increase or the absolute toughness values of cancellous bone is much less than that of the cortical bone, and that is due to their inherent porosity that is contained within the cancellous bone structure. Okay. Now, let us understand little bit on the bone mechanical properties, or what I have mentioned earlier, that structure property correlation. So, therefore, let me recall the structure of the bone at different length scale, and if you look at this different length scale, typically the length scale increases along the arrow. So, at the very lowest level of the structural description, let us start from right hand most of the structural description. So, you have this collagen bundle. So, this is the red block collagen bundle. So, and this collagen bundle there are some sidewise interaction is also shown here. So, the sidewise interaction is essentially by weaker hydrogen bonding or van der Waals type of bonding. So, therefore, at any given stress conditions, if necessary, this collagen bundle can slide over one over each other. So, the sliding of the collagen bundle, it is possible, and that leads to certain element of deformation in the bone structure. So, this is at the mineral particle level, this is the 2 to 4 nanometer level, this is the description. But then individual these collagen bundles, then in the fibrillar level, these are like several several of these collagen fibrils are present. Again, between these fibrils, again you have this kind of weaker bond structure. And then if you go to the tissue level description, then this is that small regions of the tissue which are like magnified here, and this is small level of fibrillar description that is being magnified here. So, now when you are applying that external stress sigma, then what will happen? The shear stress that will be generated along the bundle circumference, and that is what is being mentioned in the next slides. So, before that, let me also underline some of the important concepts. So, you have a mineral phase, that is the calcium phosphate or hydroxyapatite in this bone. So, when you put them when this assembly of the collagen as well as hydroxyapatite nanoplatelets are placed under tension, then what will happen? Calcium phosphate phase will be placed under tension by shearing stresses, and that will be transmitted through softer, elastically softer collagen matrix. And if you see that what is the typical difference in that elastic modulus values, elastic modulus of collagen is 1 to 2 gigapascal, which is like order of magnitude less than the elastic modulus of the hydroxyapatite as such. So, hydroxyapatite elastic modulus is 110 gigapascal, collagen elastic modulus is 1 to 2 gigapascal. So, because of this large mismatch in the elastic properties, it is quite mechanically it is quite feasible that hydroxyapatite will be loaded in tension and collagen will experience more like a shear stress under mechanical loading. So, there are two type of scenarios that are shown here. Scenario number 1 is that when hydroxyapatite or calcium phosphate phases, they are kind of present between the two collagen bundle here. So, this is one collagen bundle and this is the second collagen bundle. So, what is been shown here that this collagen bundle here, they will experience some kind of shear stress here, and the shear stress so that presence of the shear will make this collagen bundle to slide over each other, slide over that hydroxyapatite platelets. These hydroxyapatite platelets, although they will experience more like a tensile stress as I explained in the last slide. The another scenario is that you have the collagen bundle, and this collagen bundle, within this collagen bundle you have the hydroxyapatite platelets here which are contained well within the collagen bundle, and this collagen and the hydroxyapatite loaded collagen bundle when they will be experiencing the global tensile stress. So, there will be automatically there will be sliding, interfacial sliding, which will allow this collagen bundle 1 to slide over the collagen bundle 2, and therefore, this will have a very constant fibrillar strain which will be experienced by the hydroxyapatite which are contained within this collagen bundle. So, therefore, deformation at the ultrastructural level can be described by the two events as I explained to you in the last slide, that is, one is the frictional sliding between the extrafibrillar mineral platelets and elongation of the less mineralized collagen fibrils, and second one is the mineralized collagen fibrils past each other. So, damage essentially of the bone structure corresponds to the breakage of the sacrificial bonds. So, sacrificial bonds means these are the kind of sacrificial bonds I have mentioned here. So, these are like sacrificial bonds. So, this is the breakage of the sacrificial bonds and restructuring of the material under load. Okay. So, damage again this damage, so this breakage of the bonds has been explained here in more specific terms, that is, you have the non collagenous proteins and extrafibrillar mineral between the fibrils and between collagen and mineral within a fibril and restructuring of the material under load. And also there is localized clusters of such breakage, and that can lead to that can grow and link up as in conventional fiber matrix fiber reinforced composite materials. So, in a way, from the basic description of the different scale of this bone structure, it should be apparent to you that natural bone can behave more like a fiber reinforced composite. Which is the fiber component here? So, this fiber is your collagen, and what is the reinforcement here? And reinforcement is essentially hydroxyapatite or the mineral phase. So, any fiber reinforced composites in that conventional material science, you know that it shows much better mechanical properties compared to individual constituent. So, if you extend that similar kind of idea here in describing the bone properties, it should be clear to you that because that collagen fibers are dispersed in this bone structure and these hydroxyapatite platelets are equally present there. So, hydroxyapatite platelets, presence of hydroxyapatite essentially reinforce the elastic properties of this bone structure. So, I hope that I have covered both these bone structure and their properties, and to some extent how these fracture properties can be measured experimentally. Now, we will start with this some of the key concepts of the biomaterials and essentially the definitions and certain discussion of some of the concept. So, the aspects of the biomaterial science essentially deals or essentially integrates the concepts of the two widely or two remotely linked disciplines. One is that biome is biological sciences and materials being material sciences. So, these two disciplines, they are integrated and their ideas and their concepts are being drawn together to build up this discipline of these biomaterials. So, this is like more formal definition of these biomaterials. So, any material, natural or synthetic, constituting the whole or part of a living structure which performs, enhances or replaces the natural function of a tissue without evoking any undesired toxic reactions can be called as biomaterial. So, there are certain things that needs to be underlined here. So, one is the non-toxic, second one is the non-immunogenic, third one is non-thrombogenic, fourth one is the non-carcinogenic and non-irritant material. So, therefore, one has to assess each of these properties, like how one can quantify or assess the non-toxic properties or non-immunogenic properties of a synthetically fabricated or synthetically produced material. The second definition, which is much more recent, and this is given by David Williams, and which states that a material which is engineered to take a form which alone or a part of a complex system like a drug delivery system is used to direct by control of interactions with components of living systems, like living systems means cells, proteins, bacteria, blood, etc., the course of any therapeutic or diagnostic procedure in human or veterinary medicine. So, one of the core property of biomaterials, which has been briefly mentioned before also, that is the biocompatibility, and this is again a textbook type of definition. So, which essentially states that it is the ability of a material to perform its desired function with respect to a medical therapy without eliciting any systemic effects or local effects in the recipient or beneficiary of that therapy, that is human patient, but generating the most appropriate beneficial cellular or tissue response. Most beneficial cellular or tissue response, that means this is application specific biocompatibility property. So, in other words, biocompatibility definition wise is more application specific. The endpoint objective is that material, that biocompatibility property should enable clinically relevant performance of that targeted therapy. So, this is a targeted application specific, and it should involve both in vitro as well as in vivo experiments. In vitro means the experiments which are conducted in the lab with glassware or petri dishes. In vivo means experiments were conducted using animal models. So, in vitro, the Latin word means test tube, culture disk or glass, and it is an artificial environment outside a living organism such as a test tubes. Essentially, you are simulating the living system in a test tube or in a glassware, and these are the scientific experiments which are essentially performed with cells or biological molecules like proteins and so on in physiologically simulated environment, but outside the normal biological context. So, therefore, since these are the experiments which are conducted outside the normal biological context, it cannot, although it wanted to, simulate that exact environment which is present in in any animal or human patient. However, it is the first study, it is the first set of experiments which are to be conducted before one can go for the in vivo experiments, which are again defined here as the experiments conducted inside living organisms to simulate physiologically and functionally similar microenvironment around a biomaterial in relation to its targeted application. So, therefore, whenever one has to do this in vivo experiments, there you have to have a different kind of defect model or different kind of animal model to simulate the in vivo environment, to simulate the in vivo experiments. Now, specific animal model, the rationale of choosing specific animal models is also disease specific. For example, rabbit models are used more for conducting bone replacement materials, while sheep model are used more for cardiovascular implants. So, therefore, for cardiovascular implants, one is to use much larger sized animals like sheep or pig model. Okay. This third one is that host response or foreign body response. So, it is the reaction of a living system to the presence of a foreign material in vivo. So, to start with, one has to remember that whenever you implant any biomaterial in a living system, it always causes some kind of local or systemic inflammatory response, and this response in scientifically is known as the host response, and it occurs irrespective of the way that you implant the material, like either through surgery or through injection, as all biomaterials are expected to cause a disruption in the local tissue environment. Cytocompatibility also has been mentioned before, that is, it is cell level compatibility. So, essentially ability of a biomaterials to be in contact with proliferating cells without producing an adverse toxic response. and this toxicity is quantified using using different assays. Most popular assays that people use is MTT assay, and a complementary assay also one can use this LDH assay. Now, each assay, one of the things that in cell biology one has to remember that each assay actually gives you certain specific information. One cannot make any overstretching statement based on the results of one single assay. For example, MTT assay tells you the number of metabolically active cells. So, if that X percentage cells are metabolically active in a given cell population, one cannot say that 100 minus X% cells are dead, simply because in order to quantify the dead cells, one has to use that LDH assay. So, this LDH assay, when you do LDH assay, then only you can say yes, Y percentage of the cells in a given cell population is dead, it is not alive anymore. X plus Y can be 100 or may not be 100 or less than 100. So, in cell biology, one cannot kind of make overstretching statement based on the information obtained from a single assay. Chemocompatibility, hemo means blood, chemocompatibility essentially means that blood level compatibility, and it is a study of a compatibility of a synthetic material with blood and blood cells. Now, this level compatibility is important for cardiovascular materials like cardiovascular stents, pacemakers, cardiac patches and so on, and it depends on the different factors like hematology, a study of red and white blood cells, and coagulation, that is the platelet adhesion and leukocyte adhesion and fibrinogen adsorption, so which also takes place on artificial material. Ideally, hemocompatible, a perfect hemocompatible material should not cause any platelet adhesion and should be non-thrombogenic in nature. In other words, it does not disturb the delicate hemolytic balance between the coagulation and fibrinolysis. Okay. Now, having given this all these different definitions, there are two things often people are confused in these biomedical applications or as far as the biomedical materials are concerned. One is called scaffold and another is called implant. Now, these two things need to be defined with respect to their with respect to their characteristics which distinguish themselves. Scaffold normally means that is a porous material, and this porosity is very helpful for the cell colonization and cell growth and proliferation, and also to some extent cell differentiation. So, these scaffold structures are essentially 3-dimensional synthetic porous structure, as has been mentioned very clearly in the first line itself, to facilitate tissue formation in vitro. And this kind of interconnected porous structure which is present in many of the scaffolds which are experimentally studied in different research groups around the world, these interconnected porous structures are helpful for the osseointegration property, like when you implant this scaffold structure in any animal model, that also helps in the helps in angiogenesis like blood vessel formation through this porous structure, that also helps in the bone ingrowth into the porous structure. Now, those things will describe it much more later, but at this point you must understand that scaffolds usually share most of the ABCD properties or at least few of these properties. These allow cell attachment and migration. Now, what is cell migration? I have explained to you before. That is essentially motility on a material substrate, deliver and retain cells and biochemical factors, enable diffusion of vital nutrients and expressed products, and exert certain biological and mechanical influences to modify cell behavior. Now, typically the scaffold term implies porous constructs with interconnected pores more in like 10 to 100 micrometer, which facilitates tissue ingrowth, reduce limitations due to diffusion of nutrients and so on. So, the morphology and porosity are the primary importance in scaffold, whereas mechanical properties are of secondary importance. As I said, since it is a synthetic porous structure, 3-dimensional porous structure, this cancellous bone, you remember that cancellous bone also has a very large porous structure, and cancellous bone are mechanically inferior than cortical bone. Similarly, since scaffolds are essentially 3-dimensional porous structure, scaffolds also have weaker mechanical properties. And these scaffolds are important in tissue engineering. Before defining other terms, let me also explain that what is meant by implant. Implant is a general term used to describe any object that may be placed in direct contact with living tissues. That FDA, that is US body, that is Food and Drug Administration, defines medical implants as devices or tissues that are placed inside or on the surface of the body. So, it is used, many implants are prosthetics, and this is intended to replace missing body parts, and other implants deliver medication or monitor body function. Those things we are not, that does not come under the purview of this present course. However, these implants by definition, as you see, that these are like medical devices which are placed in direct contact with living tissues. Examples of the implants is THR, like total hip joint replacement. You have a stem that is made of either titanium or stainless steel, that is the stem. You have a femoral head that is either made of the steel or alumina, and you have acetabular socket, again that is made of that polymeric materials like ultramicron polyethylene. Now, all these 3 materials essentially at the bulk level, they are not 3-dimensional porous structures which a scaffold is, but that is this THR, which is a very well known example of an implant. It is essentially a 3-dimensional solid structure, non-porous structure, which is intended to carry lot of mechanical loads, as well as is intended to function under a large number of fatigue cycles during that mechanical loading conditions. So, therefore, implants are by definition meant for more like mechanical load bearing applications. Another examples of the implant is total knee replacement. So, total knee replacement also has load bearing materials. So, these load bearing materials are essentially non-porous and solid materials like total hip replacement materials. So, certainly the implants must have much better mechanical properties than scaffolds, but with uncompromised biocompatibility properties. Why implants are necessarily to have, or it is meant to they are meant to have better combination of mechanical properties with uncompromised biocompatibility properties, because biocompatibility properties, that is the bottom line for performance of any biomedical applications, and therefore, we need to understand that what is the distinction between the scaffold and implant, and I hope that in last 2 slides I have made such distinctions little clear in your overall understanding.